Hey, Teeny, welcome in. Okay, let's see. All set. There. Hello, welcome again to MicroCinema 4K. I'm Jason. Glad to see some familiar faces in the chat. Hey, Marcos. Hey, Teeny. Hope everybody's doing good. I just got back yesterday from a pretty long drive down to Southern California, the Mojave, as it were. Went out to the high desert to meet an old college friend. Anyway, that's my excuse for today. I did not go very far to collect my samples, but I actually did collect from some new places. Hey Andy, welcome back to the chat. Been a while since seen you in the chat. Hope things are going well. And today I just kind of went a few blocks away. So you guys might know, if you've been here before, that I have a stream that I'm pretty fond of. But sort of flanking that stream are several man-made ponds in certain areas. And these are generally known as recharge ponds. And so, as some of you may be aware, California has a lot of issues with water. Actually, we don't have issues with water. We have issues with not having water on a pretty regular basis. So we have these things called recharge ponds that are basically just places where we can kind of pool water up and let it slowly return to the aquifer, so it doesn't just run off all the concrete that we put all over Silicon Valley, because we're so industrious over here. Some of these recharge ponds are pretty nice little, like, kind of parky areas. A lot of ducks and whatnot, and microbes as well. So we will see some of the critters today that are living in the water that eventually would make it back to the your faucet. If you're in California, of course, it would go through a whole process to make sure it's nice and pure. So don't freak out. I know some people are always worried about, is this stuff in my drinking water? No, your drinking water is very boring, just water. So anyway, okay, we got Sydney from Australia. Hello. The bobsled, hello. And I guess we'll start off as usual with our prepared slide here. I wanted to mention I have a I'm trying out something new tonight. You guys know I like to try out some new stuff. I prepared kind of an extra deep slide. I'll talk about it a little later when we get to it. But it should be good for like some low mag stuff. And so I figured let's start off with a prepared slide that is good for a low magnification lens. And so here we have a slide from my collection of Friedrich D. Nelt, who was a German immigrant to the United States in the kind of mid 1800s, and This slide, I don't know the exact date on it, but it's in the ballpark of around 150 years old, which is pretty cool. So without further ado, we'll put me away, and we will get back to the slides. So here we go. Test 1, 2, test 1, 2, looking good. Okay. So this is a slide. Like I said, it's around 150 years old, by Friedrich Dienelt. It could be more on the order of 120 years, or it could be a little older. Kind of depends. You know, again, there's no date on it, but you can see that the mounting here is certainly aged over time and is degrading a little bit. The mountant itself is a little bit yellowed, which happens over time, but all in all, the specimen in the middle is in good shape. And I'm actually going to put a link in the chat for those of you that are watching. If you want to read up a little bit more about Friedrich Dienelt, and you can also see, this is a website called microscopist.net. It's run by Dr. Brian Stevenson, who I guess it's just kind of his hobby. He likes to catalog the old slide makers. And he's got a nice long write-up on Dienelt, and you can see some of his slides. And actually, you'll see a few just kind of in the first figure that are pretty representative of something like this slide. He was really good at mounting like large specimens, big showy stuff. So now this one, all it says is Hemiptera. There's nothing more specific than Hemiptera, and the Hemiptera are the true bugs. So this is a true bug, and that's about all I can say about it. I'm certainly not an entomologist, but from kind of tip to tip here, it's around 2 centimeters long, and yeah, it's just a pretty cool mount. And we are using a 1x objective lens here. Combine that with my 1.67x kind of projection eyepiece, and so the magnification on my camera sensor is just 1.67x, so just a little bit larger than reality. And we can zoom in. This is my 2x objective here. And it's kind of interesting when you use these super low magnification lenses. It's like the field iris kind of just functions like your normal iris does. So I'm not actually moving the condenser iris, I'm moving the field iris here. I'm using just a low magnification condenser. You see this thing's got a pretty interesting little appendage here on its backside. And there's some little hairs. There's a little blue hair that's stuck in there that's in the mount. And a little red fiber there. So not a perfectly clean mount, but I'd imagine it's pretty difficult to get something completely devoid of any contamination or debris. And you can see the eyes, see the compound eye structure in this thing, as well as its proboscis. So I think like stink bugs or true bugs. That's one. I think like a lot of the most common insects you might think of are not actually true bugs. Let's see, let's pop this up. Now I'm at the 4x lens, which should allow me to get a decent field of view here. this thing down to actually, yeah. So now when I'm using a flip top condenser. So when I'm using the 4x lens, I actually flip the top up. Yeah, get a nice flat field here. And you can see those eyes. Those eyes are really interesting to look at. They're just so neat. But yeah, I mean, as near as I can tell, I mean, this guy Friedrich, he made a lot of slides. You know, this was really his job, and I think you could get them for like a dime or each or something like that, maybe a quarter. I paid a little bit more for these, but it's always just really cool to have, you know, something like this in your possession. Since you know, it's just a cool little piece of microscope history, and you can get you know, you can get good nice antique prepared slides, you know, for anywhere from like $5 to $500, depending on how interesting and unique the specimen is, how rare everything is. With the 10x lens. So Teeny asks, are insects better specimens for, I guess, so light microscopy or stereo microscopy? I'm assuming you just mean so compound microscopy versus stereo microscopy. You know, if you have some live 3D insects, a stereo microscope is probably the best way to look at them. I like looking at my beetles with a stereo microscope. But when they're all squished down onto a slide like this, I think a compound microscope is generally the way to go. These interesting little strands here that are part of the mouth appendage. And I don't know, these are for dipping down into flowers or something like that. Yeah, just a neat little critter to check out. So anyway, we will we'll go ahead, we'll go back to my 1x here. Bring that down, and we'll start with a slide that I prepared. And I'll try to kind of explain how I made this thing. Okay, yeah, here we go. So essentially what this is I took two cover glasses on two sides, and I used some like Vaseline to kind of laminate them and stick them down. And I have one over there and then another stack over here. And so that creates kind of a shelf on either side that is 300 microns tall. And then I filled up the middle with some larger specimens and whatnot. Some cool stuff here. And yeah, I just kind of wanted to make sort of a deep slide that wasn't a well slide, because I often have issues with well slides. And I would say this was a moderately decent success. There were some tricky parts of it, but I'm pretty happy with it for a first try. I think maybe next time I'll try to use like tape or adhesive or something to kind of do the job of propping this up. It's a little inefficient to use these cover glasses, but anyway, this does give things like this large Planarian lots of room to swim around, and it's a good critter for our low magnification lens here. And this thing's this is a little under a centimeter long, this Planarian here. Well, let me see. Turn this on? No, I'll leave that off for now. It's out of the way. There we go. Okay, yeah. Got that. Now, one thing is I can't really get great dark field on these super low magnification lenses. We can kind of cheat, and that's by using polarized light. Oh, why is this so weird? And yeah, the Vaseline. Okay, so that's interesting. So the Vaseline is extremely bright and kind of glares from the side there. So yeah, you kind of learn through these things. I have one interesting critter here. We'll come back to it at a lower magnification. But yeah, you can check out this Planarian here. You can see like I think these are actually, yeah, I think these dust specks here are actually on my lens. Let's see if I can get those off. Normally dust on the lens is not an issue, but the 1X lens comes very, very close to the slide, and so the dust is actually pretty close to your specimens. Let's go to 2X here. Yeah, we're just getting a ton of glare from that Vaseline, so that's a good reason to use something other than Vaseline in the future. Vaseline super birefringent for reasons that are not immediately obvious to me. And this worm, in contrast, is not terribly birefringent. Let's see, so if I turn this down, okay, I can turn that down a little bit, just down. But this is pretty neat here. Watch this little critter go around. Wow, look at the I need to brush up on my Planarian anatomy, because that feature, that kind of internal branching structure really stands out here in the polarized light. All right, ciliates. Oh yeah, we got this thing. We'll definitely take a closer look at this thing. This is something I haven't seen before. We'll watch our worm a little bit here. Go to the 4X here. Let's see, let's pop this up. And that up, open that. This way down. Now we're at the 4X lens. Oh wow, look at this thing. Planarians are so thin. They're so flat. That's probably why they call them flatworms. But they're super interesting to watch. Let's turn on. Let's see how this looks. Here, I got my usual filter. That's the other thing. So when I use really low when I use really low magnification lenses, I actually need to, if I use my normal settings, I get kind of oversaturated colors, like it's overly contrasty. And so I need to set up like a secondary low magnification, like super low magnification color correction. Wow. Oh, these things are just so neat. There's a little midge larva there. Little chironomid larva. Wow, look at that. It is sparkly. Whatever that structure is. That is so cool. Oh, there's a little critter, a little Daphnia or something like that. Yeah, it's nice, you know, when usually, you know, Planarians, they're still pretty large, and so often they really don't have room to maneuver when you put them under a cover glass, and they can get kind of squished and slowed down. And so with kind of propping up the sample like this, I've given this guy plenty of room to move around unencumbered. Can flip over if it wants to. And of course, we got some nice algae here. Again, this is polarized light right now. Oh, I guess, well, I'll come back to that. So I'm going to switch in my dark field condenser. Now that I'm at 4x, I should be good to go with my dedicated dark field condenser. Go, we can just take that out and turn our exposure way down. And so now we'll get some more kind of realistic coloration on our specimens here. So this is a nice aquatic plant that I found. This is like an actual plant, it's not an algae. I just took sort of one of the tips of one of its fronds. Oh, and you can actually see, if we look real close, you can see that as many things are in ponds, in freshwater ponds, this one is covered with lots of little coconies. These diatoms, these little scale-like diatoms. There's a nice like side view of them. You can see them kind of resting on top of the leaf. And these things just spread out. And so usually if you've ever, you ever keep like a jar of pond water or something, you end up with a bunch of, kind of a brown film that's very difficult to clean off. That's usually coconies. It's tiny little diatoms that get all over everything. They grow on snail shells and everything. But one thing I really wanted to look at was this interesting little thing that I found. And look at this guy. This is some kind of insect larva. Oh, look at that. There's like a fluid inside of it. and it's like green. It's got these green kind of blobby, I don't know, things inside of its. It's sort of like got almost like a maggot-like body. It's very squishy, and it lives inside of this little thing here, which I think it built, like this little kind of cocoon here. And so this is really interesting. I have no idea what this is. Let's see, let's try to go back to, oh yeah, we're here at let's go to the 10x here. Or the 4x, I forgot that was one mag deeper. So now I can get a better look at this thing. And just like an odd little insect here. And that's it. It goes all the way back in there. Oh no, this thing is pretty darn cool. Let's see, I think I saw that it was actually kind of birefringent. Maybe we'll try to get the dark field polarized light thing going on here. And the carapaces are usually birefringent. But this, I think this little bag that it lives in also has a response. Maybe from how it's constructed. Oh, does it have, oh my gosh, nothing can escape. You guys see them? Look at that. We got some protists, some hitchhiker protists, some little sessile ciliates, maybe epistolis on this thing. So I guess it must munch on plant stuff, right? Because that certainly looks pretty green. It looks unusually green. But it's interesting that these like blobs have those little yellow inclusions too. I don't know. It's super neat, little bug that I haven't seen before, a little aquatic bug that I haven't ever come across. You got little critters down there. We've got another slide. Hey, look at that little stenter unfurling there. We've got another slide from this sample that is prepared more traditionally that we'll take a look at after this. And we'll switch over to DIC and take some super high mag stuff. This slide's a little better suited for let's see. Now planarians, I have found, do not enjoy light. In fact, they will try to run away from the light whenever you get them under it. And so you can kind of see when I first came across this thing, it sort of like curled up. It was like, whoa, what's going on? And now it's booking it. This is another drawback of using Vaseline here, by the way, is that especially little crustaceans seem to get stuck in the Vaseline. Which might be good if you're trying to study them, but I kind of want my stuff to be able to swim around freely. And some little water flea here. You know, I bet if we go back and check in on our flatworm, it's kind of going to be chilling. In fact, I could see it on the slide. So right now that flatworm is just chilling. It's just sitting on the slide. Now I'm going to go over to it right now. Here it is. Oh, wait. Oh my gosh. What's going on? Look. And now it runs away. Get me out of the light. These things just do not like the light. And they will retreat from it. So, yeah, it's always work following around the Planarians, because they will keep moving so long as you have the spotlight on them. Little midge larvae. See some rotifers poking around in there. A dog very interested in the water flea. Okay, well, we can check it out again. Even go to higher magnification here. Yeah, some of these critters definitely do entice animals. My cat especially likes gastrotrix. They have a very prey-like movement. Yeah. So yeah, just a cool way, you know. Here's a different type of water flea here. Ones that have like kind of the eyes stacked one on top of the other. But yeah, you know, I don't particularly like the super deep well slides usually. Sorry about that. Adjusting my mic. Don't usually like the super deep well slides. Just kind of how they behave. I find them kind of tricky to use, tricky to get set up nicely. And I do think that I would kind of prefer something like this method here, where I've just got kind of one even thickness of water going across the entire slide, and at lower magnifications, that's totally fine. That doesn't really impact your imaging too much. It's not good for high magnification stuff, but it's fine for what we're doing here. You can see, let's see. Okay, so the 2x I can get some okay dark field here, but I think when I go to the 1x, yeah, it's just too small. It's about the same as the 2x. Yeah, pretty much. Yeah, this thing's so cool though. I'll have to actually, yeah, I'm gonna take a screenshot here. I'll put it on what's that? What is this bug? Yeah. Subreddit. There we go. They'll have it figured out in a second. They're so good there. That's one of the best like subreddits. And got this little chironomid going here. We got some this debris. We got some Cimbella down in there. Got the stentor. Oh, hey, look at this. We have a little tiny worm, a little puppy worm. This is it's a flatworm, but it is not a Planarian. I don't know all the different worms that well. Snap something up. And here's our Planarian again. Here, it's gonna go. Gonna book it. I did a cool little experiment one time. I set up some filters such that one half of the field of view was illuminated with bright blue light, and the other half was illuminated with bright red light. And it was just kind of a casual little experiment. And basically, what I found is that the Planarians definitely are a lot more relaxed under the red light. And if you know a little something about photons and kind of how light detection usually works, not too surprising. But it was cool to try it out. That was way back in the day. We'll have to try that one again sometime. Yeah, well, I think we found more or less everything there is to see on this slide. I don't know if there's anything else that I can really do with this guy to sort of change things up. Maybe we'll give it a view in just good old right field. Just to see how that's going. And I will need to put some neutral density filters in here. There they are. On the back. And let's bring this up. So we're nice and sharp there. And open it up. Yeah, kind of cool. Oh, you can get a good, interesting coloration here on the this little envelope that it lives in. Yeah. You can see the thicker stuff, not quite as good for transmitted light unless you really crank it up to the point where things are really washed out in the background. Unfortunately, that kind of leads to, for stuff like this leafy stuff here, it looks okay because that's sort of thoroughly dense, but this thing just looks washed out. Okay, well, let's go ahead and move on to our next slide. And I think what I'm going to do is kind of do a full switch out here of all my imaging optics. I'm going to pop in my DIC phase contrast condenser, and I'm actually going to take off my objective turret and put on my turret that has my typical objective loadout. There we go. And before I do anything, we just put that in. Okay, so we'll pull this off. Get that going. Excellent. And let's take these off. Oh, and sorry, I gotta do one more thing here. Oh, let's put this here. Put this in. Actually, I gotta do two more things. And there we go. Excellent. And welcome back to differential interference contrast. Or welcome to it if this is your first time viewing the stream. Yeah, so this slide is one that is pretty thin. Yeah, pretty good. And it's made, again, from the same sample. And I just wanted to get some of the smaller stuff in a nice thin mount so we can check it out. Like, for instance, this stentor. And we've got a little rotifer as well. Oh, thing is really moving. It has been eating some diatoms, but also some cyanobacteria by the looks of it. Yeah, slow down a little bit, buddy. So stentor are single-celled organisms. They are ciliates. And yeah. They will generally attach themselves with that little anchor point at the back and stretch out into long tubes. And sometimes, oh, and we have another stentor that's sort of more in free swimming mode here, so it's retracted its little anchor point. These stentor are swimming around. They haven't found a spot that they really want to stretch out yet. They've had plenty of time. I'm surprised they're still swimming around, but it's cool to see them. Stentor are always interesting. They're usually full of lots of other stuff in the sample. Anything that can fit in their mouths. Yeah, we have a little rotifer here. Little monogonate rotifer. Oh my goodness. So that's one cell, and this thing is about 1,000 cells. Of course, the cells are a little smaller, a little more specialized. Now this could be, I'd say maybe Trichocerisa, but that's, I really don't know. There's a lot of little rotifers that look pretty similar to this. And they're not tremendously easy to identify. Here's another one. Same species or different species? Hard to say. This one looks like it has an egg. Look at that shell. Pointed shell there. Go back to my 10x objective here. We'll cruise around a little bit more. Get some nice Fragillaria diatoms there. This is a little, maybe a root, a little piece of root here. It's got some little diatoms coming off a bit. This is a larger rotifer. That's a nice one. Wow. You see that tiny little red dot? It's just got the one little eye spot there. This one actually seems like it's like the perfect size here. It's just like pinned. It's not overly squished or anything like that, but it's nicely held by the cover glass. I think this is I had that video a while back that got really popular on Reddit with the worm eating a rotifer, and I think this is actually one of those. I want to say that the shell looks right, looks not correct from what I remember. Actually, let's see. Okay, sorry. So I was just checking. So I think this is Euclanis. EUCLANIS, I think. Euclanis. It's rotifer. See if we can get the 40x on it. Let's put that way a little bit. Let's see what's low. Trying to find just the right spot to set my 40x lens. My 40x has a little collar to adjust to get the sharpest image depending on how deep you're focused. Obviously, it's not super easy to know how deep you're focused at any given time. And it's really meant for correcting through glass. So even if I did, it would be not super straightforward. Here's another one. Yeah, that's got quite a jiggly inside, and what's interesting too is like these little sacs in here seem to be like roiling. Like there's cilia or something inside of them that's swirling everything around. Hello. I think that's a different species. Or maybe it's just sideways. I don't know, hard to say. Oh, we got a few rotifers coming through here. This is interesting. This isn't whatever this algae is. It's not Spirogyra. Oh, check this out. Hey, these might be Vampirella amoeba here. Little amoeba that have consumed the inside of these cells. Oh, this is cool. Let's see. Let me go back to this. I'm gonna go like that. And try like that. Like that's pretty good. Maybe one more. Yeah. Wow. That's neat. And they've gone inside and are like sheltering inside. And my understanding of their kind of their little cycle is they'll eat and then they'll kind of turn into like a digestion cyst for a while. But I think that is what's going on there. Interesting. And that's the kind of stuff that, you know, I do deliberately pull stuff out while I'm looking with my stereoscope. But I don't see everything. Can't see everything with the stereoscope. So always some cool stuff to find. Some surprises to be had. Let's go back to the 10x here. And we'll kind of just do a little bit more poking around, see what else is out here. I also definitely don't remember everything that is on the slide. Oh yeah, I found some of this algae. This is interesting algae that kind of has this this sort of like hexagonal pattern to it. It sort of interlocks in this interesting way. I don't know what that's called. Oh, we got a little ostracod here. Very small one. Because it's hasn't been smushed. That one got smushed. Oh, hey, look at this. This is a neat little rotifer. Whoa. That's a weird looking one. let me get my, yeah, it's a little there. Ah. Whoa. Look at this little rotifer here. It's going to town. It's a muncher. Back at the 40x here. So this is some kind of rotifer. Ooh, oh my goodness, it can swim too. Oh, does this have, it might have a little strand that it's kind of dragging behind it. Yeah, so this is a rotifer. So rotifers are actually highly multicellular. So this thing has about 1,000 cells. They're fairly complex for their size. Kind of similar to tardigrades. You know, just small, but not uncomplex. And yeah, rotifers are almost always female as well. And they most frequently reproduce by asexual, what is it? Aparthogenesis. Where you just make an egg, and it's essentially a clonal copy of the mother. Yeah, look at this thing. This thing is busy. Very odd shape to it too. It's got that kind of little like golden backpack going on. Pretty cool. I don't know if we've ever seen one of these in particular. We've seen similar kinds of rotifers, but maybe not one of these. Knock that down. Just a little bit. There we go. Yeah. They're lovely. There's a lot of Fragillaria in this sample. A lot of nice ribbons of Fragillaria diatoms. Always very pretty. Kind of surreal to look at. Whoa, here's a nice stretched out stentor. It just hasn't found a spot. It'd be nice to see one that's anchored. I like looking at their when they finally do decide to stick to something. It's pretty interesting. Look at their backside. What do we got here? Oh, there's another one. Yeah, these are definitely the main ciliates that I was finding in this sample. Oh, there's a couple. There's a couple that are hanging out. Probably annoying each other. Oh, we got the waters receding here. Let me add a little extra water. Just pop a little drop in there. There we go. Just enough to liberate these critters. Let's spin around. Back to the 20x here. Yeah, see, I like looking at these. You can see, I guess I'm just generally sort of interested in like how these little critters are able to deliberately stick to stuff. In the center, they seem to kind of squish their butt up there, and then I guess I don't know if those are cilia that aren't moving there or something, but they have those little tendrils come out from the anchor point. And I just think it's kind of neat to understand how that's all getting done. That's a good view of the. Oh, look, there's a little diatom up, not quite small enough. Although it did swallow up a much larger diatom, as it seems. You can see they got a pretty complex oral apparatus. Sort of spirals. And it always reminds me of like either a ram's horn or like, I guess the cochlea. It's like an ear. It sort of just spirals in there, and then finally there's like a little gate, and stuff can be either sucked up or rejected. Getting bugged by this rotifer. And you can kind of see how there's like all the cilia are just sort of rushing down in there. Oh, there goes a little doodad. That might have gotten incorporated. Hard to tell. I don't see it necessarily. But these things are usually pretty efficient. And right here we can see the macronucleus. So it's sort of it almost looks like maybe like a large intestine kind of uncoiled and going down through it. But essentially that's one big nucleus. These larger, I think one of the reasons these things have macronuclei is just simply because it's, in order to get your like cellular processes done, you need to visit the nucleus a lot to get your genetic code. And so just having a big long macronucleus where it covers a lot of the cell, I guess is pretty efficient. It's not all hinged on like one tiny little spot. Hey, Mavis, welcome in. Glad you could catch it live. The other one's still down here. Gradularia. Cool. Get an idea of that razor sharp focus of the 40X. You can see there's stuff going on inside of these diatoms. They're up to something, that's for sure. There's a little Vorticella. There's a stray Vorticella. Was that a little I think. Yeah. Oh my goodness. Talk about a rough day. That's a little amoeba that is getting spun around. When amoeba are sort of floating around, they'll often kind of put little tendrils out in all directions. They look very, very curious, very alien when they do that. This one is stuck in a whirlpool. Ooh, there's a nice Cimbella. Right under the cover glass. Yeah, this diatom has kind of a shaggy covering of either some kind of fungus or just bacteria. And yeah, Cimbella are pretty famous diatoms. Just they're pretty ubiquitous, and they're quite distinct, and they have these really large poroids that you can see these little pores all over their frustules that are just very they're pretty easy to see at lower magnifications. They stand out really well. It's a nice diatom. They grow on stalks, and so this one is broken off of its stalk. I'm not sure what. Maybe that's an epithemia there next to it. We just got some other little diatoms poking around. Yeah, diatoms have, really captured microscopists ever since the invention of the microscope. they're really like perfect microscope targets, because there's almost always like more to be seen as you improve your imaging setup. Enter again. So in a lot of old microscopy books, diatoms come up pretty frequently when they're talking about the best methods to use. This is a really nice example of a stentor stretched out into its trumpet shape. Lots and lots of diatoms. Oh, I don't think it is capable of swallowing up a rotifer. Go farther down. See if we can get it to retract here. Let's see, when this thing retracts, it'll probably be like right about here. Is that macronucleus? That's all? Yeah, it is. Wow. All stretched out in like one long thing there. Maybe we won't have to. Okay. Right here. Give it a tap. You know, I have an idea. I got a plot. Let me take out my polarized light. Sorry for the white screen here. Go. Oh, we got the water coming through again. Shoot. Get that taken care of. There we go. Okay. All right. I don't know if we'll be able to see anything here. I'm gonna go to my 10x actually. Yeah. Let's see, let me close that out. Let me check my color illumination too. Brighten that up a little bit. As I set up the perfect shot. What I want to see is if there is a polarization response change when this thing contracts. So let's give it the old tappa tappa. Boom. Is it gonna... It looks like it's about to swim. I spooked it a little too much, I think. Oh, the... Yep, yeah, it just dislodged. Well, shoot. It was kind of unclear to me. I was wondering if it would get, like, a lot brighter or darker when it sort of contracts. But anyway, I guess we're in polarized light now. Always fun. Once again, we have the ongoing mystery of the red diatoms, the diatoms that are red in polarized light. So interesting. Only in certain particular sections. So strange. Okay. Let's see what else we got here. Oh, there's another picture. Maybe it's the same one. Hard to say. Little bubbles here. I did have some trouble when I was making this slide. I had to put a cover slip on it twice. Never really want to do that. Now let's see how we do with wave plate. Ooh. That's pretty hype. I should try this again. Try my little experiment again if I find another stentor to experiment upon. I'm gonna do a little something. Let's open this up all the way. Oops. I had to bump that. Go. Okay. Goes right there. Good. And then on my other polarizer. Please stand by. Okay. I'm not really getting the color split I was hoping for. Let me try again. Bear with me here, folks, while I do a little experimentation. That's very odd looking. Yeah, I guess it's interesting. I don't get the same effects. Okay. All right. In that case, I'm gonna go back to normal. There we go. Let's take this out. This way down. All right, folks, we'll get there in a second. There we go. I'll get back to here. I was enjoying this. Oh, where'd our specimen get off to? How dare you run off on me? There's one. It's interesting how I can just get it. Always so much fun. Always so much fun to play with polarized light. Oh, Marcos asked a while back if the red color of the diatoms was independent of the orientation. As far as I can remember, yes. But maybe not. I don't know. It's hard to say because, like, sometimes it's there, sometimes it's not. Yeah. It does seem to be stronger with the ones that are more diagonal here. Try, this is a little janky, but is it getting brighter and darker? Kinda. It's hard to say. It's always hard to say. I need to do more thorough investigation. Like, some species show it differently than others too. I feel like it's trying to tell me something. Huh. Wonder why I'm getting a double image there. Interesting. Well, yeah. It seems like we got most of that slide sorted out, at least. I don't know if there's anything else I want to do here. I think we'll move on to our last slide of the night. And this one comes from a different discharge pond that was nearby. And I actually found some critters in this one that I've never found in my local area before. I've only found them once before, and that is the great Volvox, the multicellular algae. And we got plenty of Volvox. Ooh, there's a nice another cyanobacteria there. We got some very large, but Volvox, when they get big, I mean, this is with the 4x lens here. Look at that thing. This is with the 10x lens. These things get big. And so these larger ones are actually like pinned by the cover glass. I'm going to go ahead and switch to, yeah, I see. Go. Now we can get a nice view of this thing, and we can see that it is covered in little flagella. I guess, well, are they, I want to say they're flagella because this thing would have come together from like flagellated algae, but maybe now they're supposed to be called cilia since the whole thing's like covered in them. But yeah, these things are present in a lot of different sizes. I found these really large ones that are easily visible with the naked eye. And then there are other ones. These are some nice chains of cyanobacteria here as well. Let's see. Ooh, there's one. Got kind of the big internal cells. I don't know. Maybe those internal spheres there become individual vorticella later. This thing, maybe it is an egg. Maybe it breaks open. But yeah, Volvox are kind of famous as being a kind of a natural step in a progression, well, kind of a final step in the progression to a multicellular organism. These would have started off as just colonies of single-celled algae. Like a rotifer kind of pinned under it. This is I recognize this. These kind of little bundles here that kind of look like little bundles of straw or something like that. This is a cyanobacteria of some kind. I forget the name. But yeah, here's like, it's interesting, like with the Volvox, like there's so many, like here's another one. And this one you can see has just like all these little individual flagellated cells. I'm kind of assuming that they're all just Volvox in different sort of states of their lifespan. But I might not be correct about that. I guess it's possible that there's different species of Volvocine algae present in this pond. But these are always just so interesting to look at because of how like orderly they are. We got, I think, some Vampirella diatoms on this algae. And a couple of ciliates here that are seem to have expired. Little strands of cyanos. That's like, this is, I think, nostoc here. This little clump of cyanobacteria. Let's see, where else? We have some more Volvox to look at here. Oh, we got a chironomid larva. And here's a good example. This is what the other cool thing about Volvox is that like it swims and like tumbles around. It's fun to watch, especially with the stereoscope. You can see these big old orbs just bumbling around. Kind of like little hamster balls or something, you know. You never know which way they're gonna go. Whoa, check this out. Oh, this is wild. Well, there's a lot going on here. So this is definitely, you know, it was a Volvox. I don't know if it still is. Look, you can see the individual cells are still flagellating here. But yeah, I think this rotifer is inside of it. And this rotifer is chowing down. Is it eating the little individual Volvox cells? Is that what its belly's all full of there? And then the other thing too is look at these little ovals inside of this Volvox. Oh, yeah, look at that. Oh, cool. See that little red dot there down near the bottom? That is a baby rotifer. This Volvox, this like husk, and it doesn't look like it's in the best shape, but this thing is full of rotifer eggs and a rotifer, which appears to be eating it. And this is not an interaction that I was aware of. I suppose if you're an algae, you kind of should expect that you're going to get munched on. Yeah, look at this thing. It's like, is it kind of sticking out from the Volvox right now? Is it trying to, did it hatch in there and is it trying to eat its way out, or did it lay those eggs? Maybe it's just been in there for a long time. Maybe it's been eating on this Volvox and laying eggs inside of it. But yeah, let's see. Is it gonna... Let's see where its mouth is. There it is. Dude, look at it. It's totally munching on these little things. If I go this way. Oh, yep, see? There they went. They went. Oh my gosh, look at it. It's slurping them down. Oh, look at that. It's eating the Volvox from the inside out. Oh man, that's wild. I had no idea that could happen. Yeah, this thing's just packed full of little Volvox cells. Wow. I'll have to keep an eye out for that in the future, like Volvox that are here to have something inside of them. Oh, it's poking its head out here. They're gonna... Oh my gosh. Whoa. That thing's fast. Oh, that's an interesting little doodad there. Oops. What the heck is that? Like buckyball. Like a testate amoeba or something? Always a mystery. The foot. I don't think I've ever had a stream where I could identify every single thing. Oh, hey, it's back. Yo. Now it's working on the outside. Dude, this rotifer is very interesting. It's speedy. Whoa. Oh my gosh, look at that. Whoa. That's so fast for a rotifer. That's really fast, especially because like some rotifers have like they can like jump really fast. They can kind of like teleport themselves by kicking some appendages. This thing's just fast. This thing just swims fast. That's crazy. There it goes. Dude, there's no way I could keep up with that. I think I know where it's going to come back to. Hello. Yep. Oh man, this thing it almost reminds me of like a little cuttlefish or something. I don't know, just the way that it's shaped. Yeah, this is what happens when you eat your vegetables. You can run around really fast. Okay, there we go with the 10x. Oh. This thing's still, it's tough. Whoa. Oh my gosh, it throws the jukes on me. Wow. Wow, this is like definitely one of the fastest rotifers I've ever encountered. My gosh. Well, that's unreal. I have to check that thing out. It's kind of just like I'm focused like halfway down right here. It's just an interesting little like video effect. They just seem to sort of pop around. Focus up a little bit here. I can make it go back and forth. Oh, that's a fun effect. Look at me. Look, I'm in control. That's neat. And sorry, folks. You know, sometimes the MicroCinema 4K ride will be a little extreme. Always bring a barf bag. What's crazy to me is that like these cells are still going. They're still, the flagella are still going. Another spinny boy. Got a little ciliate there. A little nasilid. What is that? Hard to tell if... Well... Whoa. Oh, this one's like super dense. Right there. Yeah, this one's just like super densely packed with all those little cells. Oh. Yeah, I don't know if this is like just a different phase in like the life cycle of a Volvox or what. This one versus this one. Or if they're just different species. Oh, there's a big one. Eeg Volvox. And this cyanobacteria of some kind. There's a nice amount of photosynthetic stuff. Oh, here's more of that. Oh, I wonder. This might be some kind of pollen here. I wonder if we can find any that's stuck to the top. That would be nice. Yeah, here's some. Okay. Yeah, I remember there was this kind of brownish sludge almost on the surface of the water. I did get some of it, and it was kind of interesting because I expected it looked kind of gross, and I was expecting it to smell bad, but it didn't really smell like a whole lot. And this is probably about the right color. It was kind of a deep brown with a little bit of burgundy in it. And so maybe this is some kind of just like pollen that was accumulating on the surface of the water. Okay, let's go back to the 10x. Dang. I'm still thinking about that rotifer eating that Volvox and like the eggs laid inside of it. Oh yeah, here it is, yeah. That's crazy. Anything else on this slide? Just some midge larvae. I didn't see too much in the sample other than the Volvox, you know. Obviously some rotifers here and there and whatnot, but oh hey, look at that, we got a Planarian. I always like looking at these. These are so cool. On the bottom. Let's see. We'll try to get the 40x on it. Where'd you go? Hello. There it is. Getting a good amount of ear collaboration here. I don't think I'll be able to fully get that out. That doesn't look too bad. Yeah, these things are so cool. Chlorophyllus euglenids. They like to eat stuff up. Eat up little algae, eat up other little euglenids. You probably eat that diatom. Yeah. There's a little algae. Nice diatom frustrals. I guess I found the setting pretty good here. Always interesting to see what's kind of crawling around on the bottom. High magnifications. A little bit of Melisira there. Let's see. Find that Volvox again. Huh. Oh, I see what I'm doing. Yeah, I better be careful. Yeah. I can find that Volvox without actually switching from the 40x. I don't know if I'm that good. It was near some green stuff. Oh, there's a different one. Oh yeah, look at that. So there's like the large Volvox, and then inside there's like baby Volvox. And then inside those baby Volvox, there's more baby Volvox. go back to here. Quick. Yeah, there it is. That was close. Wow. Whoa. I squished a little duder. Hey, this is one of my favorite little rotifers. This is Squattinella. Where'd you go? Oh, hey, we got a little Frontonia. The Frontonia. Let's see. There's Frontonia Lucas, and then there's another one. That's the other one. Yeah, we got Squattinella. Squattinella are awesome. These are another hit with cats, at least I found. They have a very mousy shape to them. And so yeah, these rotifers, they kind of cruise around, and on the front there, it's basically like a scoop, sort of like a manta ray has. And yeah, that scoop will just, they sort of have like their cilia, their coronas kind of built into that, and stuff flows into there as they swim around. I don't know, they're just neat little rotifers. I can get it with the 40X. You can see they have their eyes. They have two eyes, and they're pretty far offset, so they ought to have like pretty good, like, you know, binocular vision sense, which is cool. Kind of like hammerheads or something, you know. Just cool little critter. I missed these two together. Oh, hey, look at that. Hey, there's more. Whoa. All right, all right, confirmed, not a fluke. Not a rotifer that got lost. This is deliberate. These are vorticellivores. That's wild. And there's another egg in here as well. This one looks like it's working on another egg. I can't really get like a great view on them just because they're so obscured by these upper cells. But there I can kind of, there's like a little window, you can see the mouth nipping away. It's just kind of cutting away through all the, I guess there's probably like a gelatinous substrate inside of this Volvox. I think it's just trying to get to the good stuff. I wonder if that's like a specific like life cycle of these. I wonder if these are actual like Volvox specialist rotifers. They only, they hatch inside of the Volvox, eat their way out, reproduce, or just eat their way out and then go inside another Volvox and lay some eggs. If the Volvox weren't very planty, you might consider these things to be kind of horrible parasites or something. But I think we can just leave them at cute little micro animals with a very interesting way of existing. Dang, that's so cool. I'll have to post that on Facebook. See what Russ has to say about these rotifers. It's just so neat to look at. I bet that's a pretty safe place to be inside the Volvox. No flatworms coming in to suck you out of your shell, gobble you up. Dang. Oh, there's more. Wow. Here's another one. Dude, that's cool. We found something new. Who would have thought? Who would have thought that we hadn't seen everything there is to see yet? Now we probably have. That was probably the last thing. Now we've seen it all. It'll just be repeats after this for sure. For sure. That has to have been the last new thing. There's one in there too. Oh, you know, I was looking at this one earlier. Yeah, I didn't realize it at the time that this one was inside of it. It looks like it got squished. Where's that other one at? And this one. You know, it looks like, I mean... that egg is clearly, I mean, that rotifer is obviously pretty far along in its development. But I don't think it's... I have no way of knowing how far away it is from hatching. Otherwise, I might suggest we stay and watch it. It's very, very active in there, very twitchy. I don't know if I saw any indication that it was like trying to bite at the membrane or something like that, maybe. Okay, well, I think we'll call it a night. Now we did about 2 hours, which is a good solid stream. We saw some new stuff. And you know, it's always nice to like reflect on the, you know, this is just a couple blocks away in the ponds, in the man-made ponds for the Silicon Valley water table. You got some cool stuff happening. So yeah, we'll be back next week with something else. We'll figure out what I'm doing then. And yeah, until next time, everybody have a happy Halloween, and yeah, take care.